So for most of this week, we've been thinking about mammalian exon activation and how this ensures dosage compensation in mammals. But now I want to think about what we've actually learned from how dosage compensation happens in worms and in flies. So while in mammals we know we inactivate one of the two X chromosomes, and this means that you have only one active X chromosome in a female cell and one active X chromosome in a male cell, if we now consider what happens in worms you have hermaphrodites that are XX and males that have a single X chromosome. But rather than having inactivation of one of the two X chromosomes in the hermaphrodites, which would be similar to what happens in mammals, in fact you get downregulation of both X chromosomes in the hermaphrodites by 50%. So this seems to be quite unrelated to what happens in mammals. In flies, by contrast, we have the female has two X chromosomes and male has one X and one Y, just like in mammals. But now the females don't have any form of X inactivation, but rather it's the male X chromosome which is upregulated twofold. And so the way that you get accommodation for the fact that you need to have equivalent dose of X-linked genes between the sexes is that you upregulate rather than downregulating at all. So for a very long time it seemed that these three different processes had evolved distinctly, or that they were perhaps unrelated. But in recent years we've actually found out that there seem to be more similarities than we'd previously thought. So while we have this upregulation in flies of the male X chromosome, but nothing happening in the females, it's recently become clear, although it is still relatively controversial, that in mammals we not only have inactivation of one of the X chromosomes in females, but we have upregulation of that active X chromosome, which is found the single active X chromosome in females and males, just like happens in the flies. So this is relatively confusing to think about. Why is it that we should have this upregulation of the active X chromosome? But I guess what's thought is that this has probably got to do with the dose of X-linked genes compared with all the autosomes. We have two copies of all the autosomes, and therefore you get the production of protein at 2 times the rate that you would from a chromosome that only occurs in one copy. Whereas if you upregulate that single active X chromosome, you should have an equivalent dose overall of the X-linked genes compared with the autosomes. To be able to find how this occurred in mammals, it was actually needed to be studied at a genome-wide level. So you can't study a single gene on the X chromosome compared with a single autosomal gene, because the level of expression of those particular genes may not represent what happens in total. They may undergo particular tissue-specific silencing, for example, or different expression levels based on the function of that gene that's required. However, if you study all of the X-linked genes, 1,000 of them or so, compared with all of the autosomal genes, by studying the expression of all of these genes, you can then begin to see whether or not what the dose of X-linked genes is compared to the dose of autosomal genes. So as I said, this is still relatively controversial, but it appears that maybe we have something related to what happens in flies, as well as X inactivation, which we spent a lot of time thinking about. So then if there are perhaps some similarities in the mechanisms, similarities in the consequences, what about those mechanisms? What about the molecular mechanisms behind this upregulation? Well, for the downregulation that happens in worms, first of all, we know that downregulation by 50% of both X chromosomes in the hermaphrodite involves SMC proteins. These SMC proteins are structural maintenance of chromosomes proteins. And they are involved in complexes called the condensin complex, or condensin-like complexes, which when the chromosomes are actually condensed in mitosis, these are the proteins that are involved in condensing that chromatin down. So it kind of makes sense that they'd be involved in the formation of a silent or at least some repression that could be involved in this worm dosage compensation. But quite interestingly, the SMOOCH-D1 protein, which I mentioned, it seems to be involved in the maintenance of X inactivation, is also an SMC protein. It's not one of the core SMC proteins, but it has an SMC hinge domain as it's given in its name. And so there might be some relationship between the downregulation of 2 X chromosomes by half in the worms and the complete inactivation that occurs in the mammalian single X chromosome that's inactivated. If we now think about the flies, where we see the upregulation of the male X chromosome, then we know this involves long non-coding RNAs for upregulation, and these are called RNA on the X or 1, so ROX1, and also ROX2. We also know it involves histone acetyltransferases, which we know acetylation is involved in activation, and an RNA-DNA helicase, and this helicase is involved in unwinding chromatin. And finally, we know that, which has been found more recently, that the active X chromosome in the male fly is found in an active nuclear compartment. So this is like the opposite of what's found for X inactivation. So rather than creating a silent nuclear compartment, there's an active nuclear compartment that's found, which is enriched in RNA polymerase II because it's enriched in those transcription factories that we talked about in week 2. So very recently, just in 2013, in mammals it was found that also, at least in human cells, you can find a long non-coding RNA which is associated with the active X chromosome in both females and males. And this long non-coding RNA is called EXACT. This is still highly controversial, but potentially we have some similar mechanism in mammals that exists for this upregulation of the active X chromosome as what happens in flies. Furthermore, the histone acetyltransferase, which is involved in flies, if you look at the similar gene that occurs in mammals, it also seems to be occurred in mammalian X upregulation. And so, indeed, although we thought that these mechanisms were originally completely distinct and that X inactivation and X upregulation in flies were different, now it would seem that we seem to have this extra upregulation as well, and that there are similarities beyond what we first imagined. So why is it? I want to come back to this idea of why is it that we would undergo both downregulation and upregulation. So it's possible that because, as I said, we need to have not only balancing of the X-linked genes between the sexes, but also relative to the autosomes. Why it is that we'd choose to have a somewhat more complicated mechanism where we both inactivate and upregulate rather than perhaps just doing like the flies do and upregulating the single X chromosome is really still not clear. And that's going to be something that's interesting to work out why we've evolved this way, because it doesn't seem like it's the most parsimonious explanation for the way it should occur. So I'd like to alert you, we're now at the end of dosage compensation, and we're going to have one final lecture this week which is a little bit more about the fly. But I'd like to alert you to an animation that exists on this website that's given. This animation is the parental animation from which I've taken all of the snippets of animation I've shown for the first 3 weeks. This is a 13-minute long animation which goes through X inactivation, and it's really worth looking at for your own revision of what goes on. There's a little bit of additional background at the beginning on just genetics, and at the end a little bit on perhaps how it might relate to disease. So I think this is well worth your time to watch.